Hi everyone, now let us discuss on the second part of the topic that involve the process of measuring evolution of population. In this part you are going to be introduced to Hardy-Weinberg equation to solve the problems related to the topic of population genetics. Let us start with a population that is in Hardy-Weinberg equilibrium is not evolving. They analyzing allele frequencies in successive generations. Hardy-Weinberg equations are used to estimate the frequencies of alleles and genotypes in a population which is in genetic equilibrium. Okay, now let us look at the Hardy-Weinberg equation. The Hardy-Weinberg equation are representing by p square plus 2pq plus q square equals to 1. P square is frequency of homozygous dominant genotypes. 2pq is frequency of heterozygous genotypes while the q square is representing the frequency of homozygous recessive genotypes. 1 is the total number of gene pool. Let us start with the first Hardy-Weinberg equation. The frequency of homozygous dominant which is A is p times p equal to p square. For the frequency of homozygous recessive which is a is q times q equal to q square. While frequency of heterozygous which is A is p times q plus p times q equal to 2pq. Frequencies must add to 1 that equal to 100%, so this is how the Hardy-Weinberg equation which is p square plus 2pq plus q square equal to 1 are being used in counting the genotypes and allelic frequency of individual. Now let us see the second Hardy-Weinberg equation. For second Hardy-Weinberg equation, p plus q is equal to 1. P is the frequency of dominant allele which is A, while q is the frequency of recessive allele which is a. By knowing this two equation, we can use it to solve the problem related to population genetics that involve the genotypes and allelic frequency of individual in a population. Okay, from this diagram we can see how the alleles are being representing in the Hardy-Weinberg equation. Assume that there are two alleles which are A and a. Let P representing the frequency of dominant allele in population which is A. And Q is being representing by the frequency of recessive allele in population which is a. Frequencies must add to 1 that is 100%, so we can see that the total allele frequency can be said as p plus q equal to 1. Based on the diagram, as a conclusion we can say that the total dominant allele and recessive allele in the population can be represented by 100% or 1. Let us discuss in details on how these two equations are being formed. First, from this diagram, we can see that the frequency of homozygous dominant is 0.45. Frequency of heterozygous in the population is 0.2 while the frequency of homozygous recessive is 0.35. The frequency of dominant allele are being representing by both dominant allele A. The frequency is solid for 0.45. For the frequency of heterozygous, half of the allele are dominant and the other half is recessive allele. So the frequency of heterozygous is 0.1 for dominant allele and another half is 0.1 for recessive allele. For homozygous recessive allele, the frequency is 0.35 and both allele are recessive allele which is a. So, to calculate the total frequency of dominant allele in the population, we can add 0.45 to 0.1 that equal to 0.55. While to get the total frequency of recessive allele in the population, we need to add 0.10 to 0.35 which is equal to 0.45. From this calculation, we can see that the total allele in the population is 100% or 1 which come from 0.55 that is dominant allele plus 0.45 which come from the total frequency of recessive allele. So from here we can prove that the p plus q is equal to 1. Now let us see on how the second equation was made which is p squared plus 2pq plus q squared equals to 1. From this diagram we can see that the mating of two heterozygous individual from the first generation will show the combination of allele in the Punnett square. As the cross were made, half of the gamete of the F1 generation is A for dominant allele and a for a recessive allele. Once the dominant allele were combined with the other dominant allele from different gamete, it will produce the one homozygous dominant individual that represent the p squared. Next, when one dominant allele were combined with one recessive allele, it will produce two heterozygous individual representing 2pq. When the recessive allele were combined with the other recessive allele, it will produce one homozygous individual representing the q squared. So, from here we can conclude that p squared plus 2pq plus q squared is equal to 1. Now let us try to answer this question. In a population of 500 moths, 225 are homozygous dominant, 100 are heterozygous and 175 are homozygous recessive. Calculate the numbers of dominant allele A and the numbers of recessive allele A for homozygous dominant, heterozygous and homozygous recessive. Then, calculate the total number of allele present in the population. Okay, to solve this problems you need to remember, each of the individual will have a pairs of allele. So, to calculate the number of dominant and recessive allele A in the homozygous dominant population, we need to times the number of individuals with 2 to get the numbers of dominant allele A. So the answer is 450. As all the allele are dominant, so the number of recessive allele in homozygous dominant population are equal to 0. For the number of dominant and recessive allele in the heterozygous population, each of the allele for dominant and recessive are equal to the number of individual that present in the population. This is because, each of individual will carry one dominant allele and one recessive allele. So in this case, the number for dominant allele and the numbers of recessive allele, in the heterozygous population based on the question are 100. For the numbers of dominant and recessive alleles in homozygous recessive population, we need to times the number of homozygous recessive individuals with 2 to get the numbers of recessive allele A. So the answer is 350. As all the allele are recessive, so the number of dominant allele in homozygous recessive population are equal to 0. Based on this calculation, we can say that the total numbers of allele present in the population is equal to 1000. Here are some tips to answer the Hardy-Weinberg problems. First, examine the information to determine what piece of information you have been given about the population. Second, always start to find out the value of Q first. Then the value of P. If this is achieved, then every other value in the equation can be determined by simple calculation. Third, take the square root of Q to find Q. Determine P by subtracting Q from 1. Fourth, determine P squared by multiplying P by itself. Determine 2PQ by multiplying 2 times P times Q. Check that your calculations are correct by adding up the values for P squared plus 2PQ plus Q squared which equal to 1. Now let's move to the second question. The resistance to a certain poisonous compound in a snail population is controlled by a dominant allele A. 36% of the snail population in an area is resistant to the poison. To start answering the question, the first thing that you need to bear in your mind is to get the value of Q. Please be remembered that always start with finding what is the value of Q followed by the value of P. Once you get these two values, you can smile to yourself and now you can answer all the problems related to the questions. Now let's read again the whole questions. The resistance to a certain poisonous compound in a snail population is controlled by a dominant allele A. 36% of the snail population in an area is resistant to the poison. A. Calculate the frequency of the dominant allele A in the population. B. Calculate the possible number of snails with the genotypes of A, small a and small a in a population consisting of 400 snails. To calculate the frequency of the dominant allele A in the population, you need to start with finding the Q. So, you can write the frequency of homozygous recessive genotype, Q squared is equal to 0.64. The frequency of recessive allele, Q is equal to square root of 0.64 which is equal to 0.8. By using Hardy-Weinberg equation, P plus Q is equal to 1. So, frequency of dominant allele, P is equal to 1 minus Q or 1 minus 0.8 which equal to 0.2. To calculate the possible number of snails with the genotypes of A, small a and small a in a population consisting of 400 snails, we need to find the frequency of homozygous dominant genotype, which is P squared that is equal to 0.04. So, the number of homozygous dominant genotype, A is equal to 0.04 times 400 that equal to 16 snails. For frequency of heterozygous genotype, 2PQ, you need to times 2 to 0.2 and 0.8. Then you will get 0.32 for the frequency of heterozygous genotype. To get the number of heterozygous genotype, you need to time the frequency of 0.32 with 400. So you will get the answer as 128 snails. Okay, to get the number of homozygous recessive genotype, you need to start with finding the frequency of homozygous recessive genotype which is Q square. In this case Q square is equal to 0.8 square that is equal to 0.64. Once you get the frequency, you can calculate the number of homozygous recessive genotype smallest snails that equal to 0.64 times 400. As a result, you will get the answer as 256 snails. I hope that you already understand the topic. And to improve your understanding for this topic, please do more exercise that are being provided in the tutorial classes. As a conclusion, to answer the population genetics problems, always start with finding the Q-value followed by the P-value. Our next topic is expression of biological information. With that, thank you and bye.